Have you ever wondered why sometimes cells reproduce just to replace dead cells, but other times they reproduce to create life? Well, this is because there are two different types of cell reproduction. One is called mitosis, which is responsible for growth and replacing damaged cells, and the other is called meiosis, the one responsible for creating life. These two processes are commonly mistaken, so today I'm going to explain both and then I'll explain their differences. But before I do that, let's look to their similarities. Their processes consist of PMAT, prophase, metaphase, anaphase, and telophase, and their objective is to create new cells. And that's about it. Mitosis. First, there's prophase, where chromosomes are condensing, which means they're becoming thicker. Then there's metaphase, where chromosomes line up in the middle of the cell, making one straight line. Then there's anaphase, where the chromatids are pulled away and are moved to opposite sides of the cell. Then there's telophase, where the chromosomes are in opposite ends and the new nuclei starts forming to split the cell in two. Then there's cytokinesis. The cytoplasm is split, and the result are two identical cells. These identical cells are called diploids, by the way. These cells serve to replace damaged cells, and they are necessary for an organism's growth. Meiosis. The main difference that meiosis has with mitosis is that in meiosis, PMAT repeats itself, so there are two prophase, two metaphase, etc. Meiosis starts with prophase 1, where chromosomes match with their homologous pairs, which is their identical pair, in a process called crossing over. The chromosomes share genetic information in this part. Then there's metaphase 1, where chromosomes are aligned in the middle, but they are still in pairs, so they are not in a single file line. Then there's anaphase 1, where the chromosomes are pulled away to opposite sides of the cell. Then there's telophase, where the chromosomes are in opposite ends and new nuclei starts forming to split the cell in two. So far, meiosis might look very similar to mitosis, but here's where it gets tricky, because as I mentioned before, the PMAT process repeats itself. We continue in prophase 2, where chromosomes are condensing in both cells but there is no crossing over. Then there's metaphase 2, where chromosomes are aligned in a single file line. Then there's anaphase 2, where chromatids are pulled away to opposite sides of the cells. Then there's telophase 2, where chromosomes are in opposite ends and nuclei starts forming. Then there's cytokinesis, where the cytoplasm splits, leaving four non-identical cells called gametes. These cells can be found in sperm cells and in egg cells. As we saw throughout the video, the main difference between the two are first of all the overall process, seeing that in meiosis, PMAT is done twice. And then there's the purpose of creating new cells. While in mitosis, new cells are created to help with growth or to replace damaged cells, meiosis' purpose is to create life. Thank you for watching this video, and I hope you enjoyed it.